In this video, we're going to finish the topics of Chapter 6, Part 2. We first will be talking about disaccharides. And disaccharides are going to be carbohydrates that are composed of 2 units of sugar. The first one that we're going to talk about is maltose. Maltose is a disaccharide that is also known as malt sugar. It is composed of 2 glucose molecules. It is obtained from the hydrolysis of starch, and starch is a polysaccharide. It is found in cereals, candy, and is utilized in brewing in order to ferment it to make alcohol. Now, maltose can actually be found in alpha and beta forms because this molecule has an anomeric position, and I'm circling it to illustrate it. And since there is an anomeric position available, this is going to make the availability for maltose to be alpha and beta. Now, when it comes to the chemistry of how these 2 glucose molecules make the molecule of maltose, if we're going from the point of view of monosaccharides to disaccharides, let's understand that alpha D glucose paired with another alpha D glucose is going to connect to each other via the position 1 in one monosaccharide and the position 4 in another monosaccharide. When 2 monosaccharides link together to make a disaccharide, what they form is a specific type of bond that is called a glycosidic bond. And you can see that it says that is alpha 1,4 glycosidic bond. That is actually illustrating which position that carbon 1 and carbon 4 are coming together to make the disaccharide. So when we look at this alpha on maltose, this alpha on maltose is referring to the specifically the anomeric position, while this alpha is actually referring to the way that these bonds are looking at position 1 and 4. Let's now go over lactose, which lactose is actually milk sugar. Milk sugar or lactose is actually a disaccharide that is found in milk and milk products. Understand that this milk sugar can be found not only in cow's milk, but also in human milk. Lactose is actually composed of the 2 monosaccharides, beta D-galactose and alpha D-glucose. The glycosidic bond that unifies this beta D-galactose and this alpha D-glucose are specifically position 1 in the galactose and position 4 in the glucose. The glycosidic bond that forms these lactose molecules is a beta 1,4 glycosidic bond. And similar to what we observed in the molecule of maltose, lactose actually has an anomeric position that is available to form 2 anomers, one alpha and one beta. So you can have alpha lactose or beta lactose. The last disaccharide that we are going to talk about is sucrose. Sucrose is table sugar. Sucrose is obtained from sugarcane or sugar beets. When we look at the monosaccharides that come together to make table sugar is going to be alpha D-glucose and beta D-fructose. One of the things that characterizes this disaccharide is that specifically the anomeric positions from the alpha D-glucose and beta D-fructose come together to make the glycosidic bond. So as you can see, at the anomeric position, the OH in alpha D glucose is going to react with the OH at the anomeric position of the beta D fructose. So one of the things that happens is that overall, when we look at this, upon the formation of this glycosidic bond that is known as alpha beta 1,2 glycosidic bond, what occurs is that now we do not have a free OH at any of the two anomeric positions in these carbohydrates. So what happens in turn is that sucrose, out of the disaccharides that we talked about, cannot have alpha and beta anomers. Moreover, since these anomeric positions were involved in the reaction in the creation of the disaccharide, if this disaccharide is placed in a solution of Benedict's reagent, it actually cannot be a reducing sugar because it has the inability of turning copper 2 to copper 1. So sucrose cannot be oxidized, and in Benedict's reagent, since it could not reduce the copper, it is considered a non-reducing sugar. Lastly, we have polysaccharides. When it comes to polysaccharides, the polysaccharides are going to be carbohydrates that have many units of monosaccharides. In our class, we are only going to be focused on polysaccharides that have overall in their structure glucose. And you can observe here that cellulose has the monosaccharide glucose, starch has the monosaccharide glucose, and lastly, glycogen has the monosaccharide glucose. So when it comes to these polysaccharides, I'm going to talk about one at a time. What is their features in terms of the structure? You can see that most of these polysaccharides are found in plants, with the exception of glycogen, which is found in animals. As I just mentioned, they are made out of glucose molecules. You can observe that cellulose is the only one that is made out of beta glucose. Moreover, the way that these glucose molecules are connected to one another, we can observe that in the case of cellulose, they are going to be 1,4 glycosidic bonds. In amylose, we have 1,4 glycosidic bonds. In amylopectin and glycogen, we have 1,4 and 1,6 glycosidic bonds. Then, when we look at branches, meaning these are going to be side chains that are extending out of the main chain of linked carbohydrates, we can observe that when it comes to cellulose and starch, in cellulose, there are no branches. So when we look at the diagram of how these glucose molecules are connected to one another, this is how they look. Out of starch, we can see how out of amylose and amylopectin, which are the two polysaccharides found in starch, only amylopectin has branches. And that's specifically where the 1,6 bonding happens. Glycogen is also known as animal starch because when it comes to its structure, it's very similar to amylopectin. And glycogen, it is how we store sugar as humans. We can see that the shapes of these different molecules is different. It is important to note that when it comes to cellulose specifically, we can find cellulose in the fibers that are found in plants, and they are actually hydrolysis resistant. What that means is that at least for the biomolecules that we have in our bodies as humans, we do not have the ability to break down cellulose. Starch and glycogen, we do. One last application of carbohydrate that is important to note is that carbohydrates are found in blood. So in red blood cells, we have different carbohydrates that are exposed on those cells. And there are 4 blood types, A, B, AB, and O. So these blood types are actually based on 3 to 4 monosaccharides that are attached on the membrane of red blood cells. And each blood type has the monosaccharides that you see below, which are D-galactose, L-fucose, and N-acetyl D-glucosamine. So we can see actually how, at least here, we have an illustration for the monosaccharides that are going to be linked to one another, and they're found in blood types O, A, and B. You can observe that all of them have specifically fucose, which is represented by this red triangle. They're going to have N-acetyl glucosamine, which is that blue triangle. And they're going to have galactose, which is going to be this yellow circle. When we observe overall what's happening, we can see that the difference between the A blood type and the O blood type is that it has an additional monosaccharide that is specifically N-acetyl galactosamine. And for the B type blood type compared to O, then we can observe that it has an extra galactose monosaccharide attached to the linkages of carbohydrates that are exposed. And as you can see, carbohydrates are very important overall structures in the body.